So once we talked about the antibodies, this is a perfect idea to start talking about the plasma cells right now. So the plasma cells are what? They are specialized, differentiated B lymphocytes that have one very important function: to produce antibodies. Of course, this is a discussion best ideally spoken to and discussed in the immune system and of course immunology, but just a short discussion right here. Of course, we have any molecule that is seen as foreign from the human body is considered to be an allergen, an antigen. So whenever there's an antigen induced and placed in the human body, there's a whole immune reaction, the adapt immune reaction, which is going to produce specific and special antibodies against this one unique antigen. Generally speaking, there are two portions and two fragments that we should discuss and we should know when we talk about antibodies. There is the FC portion. This is the constant portion. This doesn't change ever. And the FAB, the F antibody fragment. This is where the unique portion, the unique, let's say, construction, the unique conformation of the antibody is placed there. So, of course, this is the area where we can bind, where the antigen is going to be bound, exactly in this FAB fragment. So the plasma cells have this unique clockwise appearance, which is the circular, very nice spherical nucleus along with a high amount of cytoplasm. So let's talk a bit more about the cells of the connective tissue that we can find in the white blood cells, the leukocytes, and which of these can be found in the connective tissue. So we have, again, in the discussion of blood, we'll see the whole category is much wider, but in this case let's just focus just a bit on the two different stem lines that can be produced by the blood stem cells. So we have the lymphoid and the myeloid. Now, as a result, after many steps of differentiation and many steps of different stimuli being exposed and exposing these cells in different stimuli, we're going to have the production of a wide range of different cells. So first, let's discuss about the natural killer cell. This is a cell that it typically aims and typically destroys and kills human cells that are infected typically with a virus or some other elements of either cancer cells, premalignant cells. Then we have the T lymphocyte. These cells are unique cells of the adapt immune system, the specialized immune system, and they are typically there are many different forms of the T lymphocytes. Namely, we have the CD4, the CD4, the CD8, cytotoxic NK T cells. And typically the function, of course, is to produce cellular immunity. And we have the B lymphocyte, which transits into the plasma cell. In this case, of course, this is the humoral immune reaction, humoral defense, and this is the production of antibodies, which are placed, and these antibodies are typically placed in the lymph or are typically placed in the bloodstream. So, of course, in the myeloid stem cell, we're going to find the different types of eosinophils and granulocytes. These cells are discussed thoroughly in the blood, along with the platelets and the red blood cells. These cells, again, are discussed in the video of the blood. So, to sum up, these are the cells of the connective tissue we discussed so far. Fibroblasts, again, the cells that produce fibers and ECM and extracellular matrix, or the plasma cells that produce antibodies. We have the adipose cells, the two different types, the brown and the white. The brown, the primary function of the brown is to produce heat, thermogenesis, and that of the white is to function as an energy storage, and also as an insulator to protect from mechanical and thermal sources and stimuli. We have the lymphocytes. We discussed right now the B and T lymphocytes. Just shortly, of course, there's going to be a much more longer and extensive discussion in the immune video. And then we saw the macrophages, and specifically the mononuclear phagocyte system, which of course is composed by the monocytes and the many different forms that they can transist to and they can actually, let's say, adapt into. Then we saw the fibrocyte, which is the quiescent, the almost inactive form of the fibroblast, which is much shorter, much thinner, less amount of cytoplasm, and of course shrink to nucleus. And the eosinophil neutrophil and these two cells are discussed thoroughly in the blood. Shortly, eosinophils are responsible for the reaction and the defense against helminthics and parasites, and helminths and parasites. Neutrophils are typically cells that perform the function of phagocytosis, along with many other unique features. For example, the extracellular neutrophil traps. This is again something beyond this point of the discussion right now. And lastly, we'll discuss about the mast cell. Of course, the mast cell is the cell that is responsible for the allergic shock reactions and general allergic reactions. And we find in these cells these two different molecules, heparin and histamine, which we should absolutely know and be familiar with. So now let's move on to the ECM. This is, of course, the content that is typically produced by fibroblasts and other cells of the connective tissue. The ECM is composed out of two different contents, the fibers and the amorphous ground substance. Let's go on to it. Let's go see what is which. First, we're going to talk about the collagen. Collagen is the most commonly found fiber in the body. The highest amount of fibers in the human body is collagen, of course. Now, collagen is typically produced by a triple helix of alpha chains. What are the alpha chains? This is the chain that's being produced by the cell, and we'll discuss the whole procedure, how it works, but at this point just understand that this is a triple 3 alpha chains entangled together. Now, collagen is being produced in the human body constantly and requires vitamin C to do this procedure. And there's also this constant rate of destruction and production of collagen. This procedure is called remodeling, typically speaking. So we're going to find the MMPs. The full name is matrix metalloproteinases, and these are unique enzymes along with collagenases. These are enzymes that are being produced by typically the macrophages or neutrophils or many other immune cells that typically destroy and cleave and tear apart the collagen in the human body. And of course, this collagen again is being produced again from the fibroblasts and fibrocytes and a couple of other cells. So let's talk a bit about the alpha chains and how does this whole production of the collagen works. Well, first off, we have the production of the alpha chains in the RER. In the RER, we're going to find the first assembly of the pro-collagen molecule. This is simply three molecules, 3 alpha chains that are being produced in the RER, and then they're being processed in the Golgi apparatus. So these triple helixes, these 3 alpha chains come together, aggregate together, there's an assembly of these 3, and this unit of the 3 alpha chains is called pro-collagen. Why pro-collagen? Because this is not the fully complete structure of collagen. So these pro-collagen molecules are being stored, adapted, and modified in the Golgi apparatus, and after this procedure we have the exocytosis, the exocytosis of these molecules. So the pro-collagen is being produced and is being released outside in the interstitial space. And in this case, after the production, after this exocytosis, the fragments, these many different units of pro-collagen, are going to be bound together through cleavage. How? We have on the both ends of the pro-collagen molecules, we're going to have these non-helical propeptides, these small, let's say, caps in both sides, which will be cleaved with the function of pro-collagen peptidases. This is what we discussed before. So they were cut exactly in these portions and the 2 portions from the both ends, and allow the many different pro-collagen units to be bound together to actually form the collagen. Collagen typically can be formed into fibrils. The first step is, of course, the formation of a fibril. And now they're based on a different type of collagen, because we have many different types. Over 20 different types of collagen in the human body exist. Some of them form just fibrils, so this is just the cleavage of the pro-collagen unit and the formation of collagen, and that's the end of it for some types of collagen. But in other types of collagen, for example collagen type 1, we're going to have the aggregation of many collagen fibrils together to form the one collagen fiber. This is typically what happens with type collagen 1. And in other cases as well, we're going to have many collagen fibers come together and aggregate together to form the bundle of the collagen fibers. So the bundle of collagen fibers is composed out of collagen fibers, many collagen fibers joined together. The collagen fibers are formed by many collagen fibrils coming together. Then we have the reticular fibers. Now the reticular fibers are unique fibers that can be found typically in few organs, in few locations of the human body, some in low amounts typically throughout the whole human body. And the term reticular fiber is practically used because the name reticular means network. Okay, so because they form this networking appearance, this is why they're called reticular fibers. And also we have these fibers are produced by reticular cells. Don't be fooled. This is just a very fancy term to denote that the reticular fiber is just collagen type 3. That's it. It's an agariophilic, which is silver which stains positively in silver impregnation methods. And in this case, of course, we have the reticular fibers. The reticular fibers are collagen type 3 specifically, and they're produced by specialized fibroblasts, which are called reticular cells. So again, the reason why we have this different name is, of course, because there's a tiny different function of these cells from the fibers, and of course that there's this unique feature that they can be found in typically in immune organs. For example, the lymph node. So next up we have the elastic fibers. The elastic fibers are they're composed out of two different molecules, very important: the macrofibrils and the elastin molecules. The macrofibrils are the long fibers on which the elastin molecules are going to be attached, and they're going to cross-link between them. And because of this cross-link of the many fibers and the many elastin molecules, they're gonna form this conformational shape and size, which is more, let's say, resistant to strain, to pulling motions. Typically found a high amount of elastic fibers in the tendons, for example. Now let's focus on the amorphous ground substance. In simple words, amorphous means that has no shape. Amorphous, that's what it means, of no shape. But the point and the functionality and the function of the amorphous ground substance is to form the pillars and to form the scaffold, the matrix in which the collagen, the connective tissue, is going to be formed. So there are three elements we should discuss first, and we're going to see a bit about all three. First, we're going to find the glycosaminoglycans, GAGs for short. Then the proteoglycans and glycoproteins. Just a small help, whenever you hear the term proteoglycans or glycoproteins, to understand what are they made of majorly, and what is the major portion of this molecule and what is the minor portion of this molecule. Typically the first prefix is the one that is that of the higher amount, and the second is that of the lower amount. For example, proteoglycans, proto is the first, protein means proteins, so there is a major amount of proteins and a smaller amount of saccharides attached on them. In the glycoproteins is exactly the opposite. The saccharides is the major part, and the proteins is the smaller amount that we can find. So let's focus first on the glycosaminoglycans, the GAGs, also called mucopolysaccharides. This name, you should have it somewhere lodged in your brain, because their diseases, metabolic diseases, called mucopolysaccharidoses, and of course their metabolic disease is based on the malfunction and the malformation of these units, of these amorphous substance parts. So glycosaminoglycans, of course, are just nothing more than T-saccharide polymers. Of course, this means that there are two different saccharides or same saccharides in repeating motion after and after and after, in many repetitive sequences. So this is the structure of the glycosaminoglycans. They can be bound to other proteins, typically found in the hyaluronic acid, and there can be also unbound, and also bound to other proteins, for example, the keratin sulfate. We'll discuss the functionality along with the proteoglycans, because this is the perfect opportunity to bind them together, and you'll see why they can be easily explained together. So let's go to proteoglycans. As I said before, proto is the core, the highest amount of the proportion of the molecule, so majorly proteins, and then the glycosaminoglycans, which are saccharides, the part of the glycans part. So let's see actually let's see in practice how this actually works, and what's the point of what's the function of these proteoglycans. So let's take for example one of the most well-studied proteoglycans, the agrican. So the agrican is constructed with a hyaluronic acid axis. All of this is the hyaluronic acid, all of this, And now on this hyaluronic axis we have bound glycoproteins. Again, this is the glycoprotein part, and this is again the glucosaminoglycans. So they form this whole, let's say, complex of hyaluronic axis, the link protein and core protein, and along again we have the glucosaminoglycans. This is the whole conformational size. The whole architecture is actually very complex. And what's the point of this? Well, the point is that because we have many spaces, many small spaces, and specifically they can form, let's say, this gel-like appearance. This is where we can actually store water. This is where water is being stored in high amounts, and also we have a high amount of growth factors within this whole matrix of the aggregan. So the point and the function of the aggregan, and as in consequence most of the proteoglycans, is that they produce this physical properties of flexibility, of resistance to strain, to stress, and they provide mechanical properties by practically abusing water, by enclosing water in a closed area and exploiting the physical properties and mechanical properties of water. This is the first function that we should actually understand when we see aggregan and generally proteoglycans. This is the first. And the second is that because they have this high amount of water, this is also where they have the growth factors inside them indispersed. The question is why? Why do we need growth factors exactly in this area? Well, this is because generally speaking, the connective tissue is undergoing continuously a procedure of destruction and production. This whole procedure of remodeling, a non-stop remodeling procedure. And in this case, of course, we're going to have when the destruction of hyaluronic axis and the whole structure of the aggregan is inevitably in the area, is inevitably in the steps, then these growth factors will be released. And because of this release of the growth factors, we're going to have the potential for the other cells to receive the stimuli, which are from the growth factors, and start reproducing again, start producing the whole structure from the beginning. Lastly, we have the multiadhesive glycoproteins. What is the function of multiadhesive glycoproteins? Well, as the name suggests, multiadhesive, this is a glycoprotein that allows the many bindings and many adhesions between different molecules. What is the point? Well, the point is that because they connect cells to structures and structures to structures, typically cells to structures, this is how the human body, how the connective tissue instructs where what cell is going to go and what is going to produce. This is how the human body, this is how the connective tissue knows where we should have it, specifically a fibroblast, where we should have a macrophage, where we should have a mast cell, and so on and so on. So this is the maestro of the whole procedure. This is the one that dictates where goes what and where is being produced what structure. So this is again the procedure of the attachment between the ECM and the cells. So it controls cell migration and cell positioning, and as a consequence the positioning and the location of the whole ECM structures along with all of the rest of the molecules that we discussed so far. Of course, this is a sum up of what we discussed so far: the functions of the amorphous ground substance, the function of the ECM, that it binds water, also occupies space, and as a consequence we have also the mechanical properties of water. It follows the matrix for cell migration and growth. This is in part because of the amount of because of the aggregan structures and protoglycan structures along with the multiadhesive glycoproteins. And again, this is what we discussed before, that because we have the growth factors interspersed in this area in the ECM, then we're going to have, after the procedure, of course, during the procedure of remodeling, after the destruction of the structures, these factors are going to be released, and again the circle begins from the beginning. Now, this is a very important picture that we should absolutely understand, no questions asked. This is how the human body, of course, functions and stays alive. We have all the cells, of course, require high amount of nutrients and high amount of oxygen, and this is this happens through the function of the capillaries, of course. So we have the arteries that typically have high pressured volume, high pressured blood coming from the one end, and on the venule, which is a low pressured area. So of course the pressure that's being exerted from the arterial part, this is because of the heart, because of the heart pressure. This is the blood pressure we discussed, and the blood pressure is responsible for this high amount of hydrostatic pressure. What is the point of this? Well, this is the first step of diffusion. This is the first step of how we have nutrients from the blood to the interstitial tissue, to the cells, and to the tissues. So, because we have this high amount of hydrostatic pressure, because we have this high pressure system with high amount of pressure coming from the arterial end, we're going to have them movement and the movement of molecules and nutrients from the blood to the tissues. So we're going to have this constant income of nutrients to the interstitial space. So how do we take it away? How do we take away the toxic metabolites? How do we take away the toxic material, byproducts, and so on? Well, here comes the function of the blood. Because we have specific proteins in the human blood, for example, albumins, they exert this osmotic pressure. And again, the osmotic pressure is the difference between the osmotic pressure is controlled by the amount of contents and ions we have from one end, from the one liquid to the other. Because we have a high amount of albumins in the blood, the osmotic pressure is going to be higher. So the osmotic content concentration is going to be higher. So we're going to have the movement of nutrients and toxic byproducts typically, and other byproducts from the interstitial tissue to the vein. So this is how again we remove the toxic byproducts and allow the cells to be relieved of the toxic possible outcomes. There is of course an imbalance in the procedure. The difference is that this imbalance is based on the fact that the hydrostatic pressure is higher and more strong in value than the osmotic pressure. So this causes, as a consequence, we're going to have a higher amount of income of nutrients and other fluids from the blood to the tissue than the outcome, which is from the tissue back to the vein. And all of this extra amount of the interstitial liquid that's going to be produced and is going to be, let's say, encased in the interstitial space, all this excess liquid is going to be called the lymph. This is the definition of the lymph. So this lymph is going to be in time reabsorbed through the lymphatic capillary, and in the end up in the venous system, again in the veins. So this is where we find the first the definition of lymph and how this excess amount of interstitial fluid is being relieved from the tissues and is being returned to the bloodstream.